Hello, welcome back. In my previous video, I discussed various staining methods to make a permanent mount for microscopic identification and differentiation of bacteria. Today, I'll take you through the principle, procedure, and variations of gram staining. In 1884, Hans Christian Gram discovered this staining technique that could be used to make bacteria in infectious specimen more visible. Even in the presence of elaborate and expensive medical technology, the Gram stain remains an important and unbeatable first tool in the diagnosis of bacterial infections. The Gram staining consists of timed sequential application of primary dye, the mordant, decolorizer, and finally a secondary dye. We have learned that bacteria are negatively charged and they attract positively charged dyes or basic dyes. At the same time, we should note that being gram-positive or negative has nothing to do with the electrical charge of the bacteria or the charge of the dyes. It simply differentiates between the bacteria that get stained with the primary dye from those that take up the secondary dye used in the gram stain. The bacteria that retain the primary dye are called gram-positive and those that take up the secondary dye are called gram-negative. The different results with gram staining are due to differences in the structure of the cell wall of bacteria. Gram-positive bacteria have thicker peptidoglycan layer than gram-negative bacteria. The entrapment of the primary dye is far more extensive in gram-positive cells than in gram-negative cells. Gram-negative cells have an outer membrane having lipid content that get easily dissolved by the decolorizing agents. The requirement for gram staining include glass slides, wax pencil, Bunsen burner, nichrome loop, bacterial growth on media, distilled water, and light microscope. Reagents used for gram staining include primary dye, that can be crystal violet 2% or methyl violet 0.5% or gentian violet 1%. Mordant, that is grams iodine. Decolorizing agent, that can be either acetone or alcohol. Acetone is a fast decolorizer and should be used only for 2 to 3 seconds, and alcohol takes up to 1 minute to decolorize. If we expose the bacteria for longer duration with a decolorizing agent than recommended, even gram-positive bacteria will get decolorized. Secondary dye, also known as counter stain, that can be either safranin or dilute carbylfuchsin. Procedure for gram staining. First, we take a clean dust and grease-free slide and make a small circle in the center of the slide with a wax pencil. Now sterilize the nichrome loop and allow it to cool. Put a drop of distilled water or normal saline onto the sterilized loop. Transfer the drop to the center of the slide. Sterilize the loop again and after cooling, pick a small portion of a colony and transfer it to the drop of water on the slide. Spread the bacteria mixed in water or saline evenly to fill the circle. Allow the slide to dry. It may take a few minutes for the slide to completely dry up. Now pass the air-dried glass slide 3 to 4 times through the blue part of the flame with smear side facing up. The flame should touch the slide for 1 second each time. Do not overheat the slide as it can lead to charring. Turn off the Bunsen burner before proceeding to the next steps as the decolorizing agent is highly inflammable. Bacteria are naturally colorless and they all look alike under the microscope. The primary dye is added to cover the fixed and dried smear of bacteria and allowed to stay for 1 minute. Wash with water to remove the excess stain. The primary dye is taken up by all bacteria. If you observe the slide under the microscope, you will find all bacteria stained with the same purple color. Now we add a mordant, that is Gram's iodine. It is a stabilizer that causes the dye to form large complexes in the peptidoglycan meshwork of the bacterial cell wall. Wash with water to remove the mordant after 1 minute is over. Next, we proceed for decolorization of the bacteria. This can be done by using either acetone or alcohol. Acetone is a rapid decolorizer and should be used only for 2 to 3 seconds, whereas alcohol is used for 15 to 30 seconds till the slide is completely decolorized. The alcohol dissolves the lipids of the outer membrane of bacterial cell wall and removes all the primary dye from the bacteria that have thin peptidoglycan layer. By contrast, the primary dye is tightly embedded in the bacteria that have a thick peptidoglycan layer that is relatively inaccessible and resistant to removal by alcohol. You can see that some of the bacteria have become decolorized by acetone. Wash with water. The decolorized bacteria will not be visible under the microscope, so we add a secondary contrasting dye. The dye used is either safranin or dilute carbulfuchsin. We allow the dye to stand for 30 seconds. You can see that the decolorized bacteria have now taken up the pink color of the secondary dye. Finally, wash the slide with water and dry it using a blotting paper. The slide is now ready to be observed under microscope. You should be able to differentiate the purple gram-positive bacteria from pink gram-negative bacteria. In this slide, you can see that we have divided the common pathogenic bacteria based on the gram reaction into gram-positive and gram-negative. We can also use gram staining to identify various shapes of bacteria such as cocci, bacilli, vibrio, and spiral. Gram staining also helps us find the common arrangements of bacteria that can be single cocci, diplococci, capsulated diplococci, cocobacilli, single bacilli, grape-like clusters of cocci, example staphylococci, cocci in chains, example streptococci, cocci in tetrads, bacilli in palisade arrangement, diplobacilli, cocci in sarcina arrangement, and bacilli in chains. Gram-positive organism can appear as gram-negative in the following scenarios. Over-decolorization of the smear. Smear is prepared from old cultures. Use of old iodine solution. Cell wall damage of the bacteria due to excessive heat fixation. There are certain bacteria that do not colorize with gram stain. For example, Mycobacterium tuberculosis. It has too much lipid in the cell wall and primary dye cannot penetrate. So we perform acid-fast staining to visualize this. Treponema pallidum is too thin to be seen under bright field microscopy. So we perform dark field microscopy or fluorescent microscopy. Chlamydia and rickettsia are intracellular organisms. So to visualize them, we need to perform Giemsa stain or any other tissue stain. Legionella pneumophila has a poor uptake of the counter stain. So it has to be visualized by prolonging the time for the counter stain. Now there are certain variations of gram staining. Based on the decolorizer used and the secondary dye or even the variation in the time of application of these reagents can give rise to modified form of gram staining. Some modification of gram stain are shown in this slide. So this was all about gram staining. Hope you liked the video. Do comment, like, subscribe and share for more of such videos. Thank you for watching.